Good morning, everyone. Welcome to day 3 of the October 2020 ACIP meeting. Today's meeting will be dedicated exclusively to COVID and COVID vaccine. We'll begin here shortly. I'm going to take roll from the voting members. I will call your name. You do not need to give your affiliation or whether you have any conflicts at this time. So I'll just run through the list. I'll start with myself, Jose Romero here. Dr. Robert Atmar.
Present.
Dr. Kevin Alt.
Present.
Ms. Lynn Bata.
Present.
Dr. Beth Bell.
Present.
Dr. Henry Bernstein.
Present.
Dr. Sharon Fry.
Present.
Dr. Paul Hunter.
Present.
Dr. Grace Lee. Dr. Veronica McNally. I'm sorry, Ms. McNally.
Present.
Dr. Catherine Paling.
Present.
Dr. Pablo Sanchez.
Present.
Dr. Peter Szilagyi.
Present.
And Dr. Kip Talbot.
Present.
Very good. So before we begin, I'd like to take chair's privilege by saying a few words before we begin our topics for today. I just want to take a few moments before we begin the COVID-19 portion of this October's ACIP meeting to make a few comments regarding the work of the ACIP over the last 7 months. Faced with a crisis of historic proportions, basic scientists, and clinical investigators, as well as volunteers, have worked with unprecedented speed to develop, test, and evaluate and manufacture a plethora of potential vaccines against SARS-CoV-2. The mandate given to the ACIP has been to develop equitable, just, and fair policy guidelines for the use of those vaccines that are proven to be safe and effective in preventing and mitigating SARS-CoV-2 disease. The volume of data that has been compiled, analyzed, synthesized, and presented to the ACIP voting members, liaisons, ex officio representatives, and the public has possibly exceeded that ever presented to any other vaccine that has come before the ACIP. It can be said without hyperbole or exaggeration that no other national COVID-19 vaccine advisory group has dedicated as much time to address the questions posed to the ACIP regarding the use of those COVID-19 vaccines. Over the 7 months since its creation, the ACIP's COVID-19 workgroup has met more times than any other non-standing ACIP workgroup has previously. I want to publicly acknowledge the extraordinary work in terms of time, effort, and quality that the members of the COVID-19 vaccine workgroup have dedicated to this effort. When I refer to members of the workgroup, I'm speaking not just of the CDC leads, Drs. Dooling, Oliver, and Mbe, or its chair, Dr. Bell, or the 3 ACIP members that include Drs. Lee and Talbot. But I'm also talking about the liaisons, ex officio members, CDC analytical staff, technical staff, and support staff, as well as the consultants that have contributed to these efforts over the many months. I would be remiss if I failed to note and praise that other ACIP voting members, other liaisons, and ex officio colleagues who, although no part of the COVID-19 vaccine workgroup, have given of their time to attend and meaningfully participate in all of the ACIP public meetings this year, including the current ACIP meeting. In the 10 months of this year, all of the current members, ex officios, and liaisons have prepared for, attended, and participated in the equivalent of 2 years' worth of open public meetings. Lastly, let me acknowledge the extraordinary leadership of Dr. Nancy Messonnier, the director of the National Center for Immunization and Respiratory Diseases, our ACIP executive director, Dr. Amanda Cohn, the assistant executive director, Jessica McNeil, for their support and leadership in this difficult task. Lastly, with regard to technical support, I think it's important to acknowledge the work of Ms. Stephanie Thomas and Ms. Natalie Green, who has provided a lot of the material that we review prior to these meetings. I thank you all for that have dedicated, all the time you have dedicated, are dedicating, and will continue to dedicate to ACIP's efforts to provide guidance for the use of safe and effective COVID vaccines. I believe that history will not forget your dedication to this task. Thank you. With that, let's begin our session for this morning. So we will talk about COVID disease, 2019 COVID vaccines, with an introduction by Dr. Beth Bell. Dr. Bell, please.
Thank you very much, and good morning. And before I begin, I just want to take this opportunity to also thank Dr. Romero for his leadership and incredible assistance and support and vision in the context of the ACIP's work in addressing the COVID-19 pandemic and working on COVID-19 vaccine policy. Could I have the next slide, please? So since as a reminder, at the last ACIP meeting in September, the ACIP reviewed an overview of COVID-19 vaccine safety, enhanced vaccine safety surveillance, vaccine implementation, reviewed some of the data around disparities among COVID-19 epidemiologic groups. We had an overview of vaccine equity and prioritization frameworks, and reviewed the work group considerations with respect to phase 1 allocation of COVID-19 vaccines. Next, please. Since the September 22nd ACIP meeting, the vaccine work group continues to meet weekly. And this is a list of the topics that we covered in October. We reviewed available information on reinfection of COVID-19, post-infection immunity. We had a number of discussions to finalize the outcomes for GRADE. We heard some analyses of modeling data for initial allocation of vaccine. We reviewed the current epidemiology of COVID-19 in pregnant women. We reviewed the ethical principles to inform initial allocation of vaccine. We heard the clinical development program for 2 COVID-19 vaccines, including data from phase 1-2 clinical trials and plans for phase 3 clinical trials. And we had further discussions regarding COVID-19 vaccine allocation. Next. Here is today's agenda, which I'll just review quickly so that everyone is aware of what's upcoming. First this morning, we'll have a section around vaccine development and regulatory perspectives with an update from the VRBPAC meeting by Dr. Fink from FDA. Then we'll hear from 2 manufacturers from Novavax, Dr. Dubovsky, and from Janssen, Dr. Sadov. In our section on implementation, there will be an update on vaccine implementation planning by Dr. Roth, and a discussion of Vaccinate with Confidence by Dr. Amanda Cohn. In our safety section, we will hear from Dr. Steve Anderson about FDA safety surveillance systems, and from Dr. Tom Shimabukuro about authorization safety monitoring plans. We will have a section on allocation and epidemiology, in which we will hear from Dr. Matt Biggerstaff about modeling strategies, from Dr. Megan Wallace with some updates to immunity and the epidemiology to inform COVID-19 vaccine policies, and a follow-up discussion from Dr. Mary Chamberlain about ethical principles for early vaccine allocation. And finally, in our section about our workgroup interpretation, we'll hear from Dr. Sarah Oliver and from Dr. Kathleen Dooling, who will discuss the policy questions and the evidence to recommendation framework and outcomes. Next, please. Now, I just wanted to briefly do another update of the status of the various vaccines. There are over 200 COVID-19 vaccines currently under development, and in the United States, there are 4 vaccines in active phase 3 clinical trials and 5 vaccines in active phase 1-2 clinical trials. Next. In terms of the vaccines that are in phase 3 clinical trials, there are 4, as I mentioned. There is AZD1222 vaccine manufactured by AstraZeneca, which announced the removal of an FDA hold on 10/23, and they are resuming phase 3 trials. The Ad26 vaccine by Janssen announced lifting of its safety pause also on 10/23, and that they are resuming their phase 3 trials. The Pfizer BioNTech vaccine, as of 10/26, reported having enrolled 42,133 participants, 35,771 of whom had received their second vaccination. And they reported that 30% of their participants in the US have, quote, diverse backgrounds. Of note, Moderna's RNA vaccine, the enrollment is now complete. They reported 30,000 participants enrolled as of October 22nd, and 25,654 participants have received their second vaccination. Next. Now, we thought it would be useful to just review the distribution, demographic characteristics of the enrollees, participants in Moderna's phase 3 clinical trials. This is taken actually from their website. And as you'll note, in terms of race ethnicity, 63% of the participants are white, 20% are Hispanic, 10% are Black, and 4% are Asian, and 3% are other racial ethnic groups. The age distribution is shown on the other side of the slide, and you'll note that there are somewhere on the order of 8,000 participants who are age 65 and above, and you can see the age distribution of the rest of the participants there. The other couple of things we wanted to draw to your attention are that 22% of the participants are healthcare personnel, and that 27% of the participants report living with comorbidities, including diabetes, cardiac disease, lung disease, and obesity. Next, please. Here is a table of human clinical trials in the United States with the 4 vaccines in phase 3 trials at the top, and then the additional vaccines that are in phase 1-2 trials below. We'll be hearing from 2 of these manufacturers during this meeting. Next, please. And this is the continuation of the slide, the list of vaccines in human clinical trials. Next, please. This is a table of vaccines in human clinical trials outside the United States as of October 19th. These are the vaccines in phase 3 trials, and as you notice at the top of each of these tables that there are additional inactivated vaccines in phase 1, 2, 3, and that there are 10 protein subunit vaccine candidates in phase 1, 2. Next, please. Continuing, these are the viral vector vaccines in human clinical trials outside the United States. These are the phase 3 trials, and there are an additional 7 non-replicating viral vector vaccine trials in phase 1, 2. And the RNA vaccine, as you can see, and there are an additional 7 RNA vaccine candidates in clinical trials outside the US in phase 1, 2. Next. Again, just a reminder of the ACIP workgroup members. And in addition, as I mentioned, to thanking Dr. Romero, I also wanted to take this opportunity to echo Dr. Romero's thanks to all of the members of the workgroup who have continued to be extremely dedicated to the task before us. Next. Here are the CDC participants. And again, I would like to join Dr. Romero in thanking them for their contributions. Next. Thank you very much, and we'll proceed with the rest of the session.
Thank you very much for that introduction, Dr. Bell. I'll next ask Dr. Doran Fink to present an update on vaccines and related biological project advisory committee meeting. Dr. Fink, please go forward.
Thank you. Can you hear me?
Yes.
Okay, great. So I've been asked to summarize a very long day of very complex discussions into very few slides. So without further ado, next slide, please. By way of introduction, the Vaccines and Related Biological Products Advisory Committee, or VRBPAC, reviews and evaluates data concerning the safety, effectiveness, and appropriate use of vaccines and related biological products. For those of you who are not familiar with the VRBPAC, it is a committee of experts external to FDA that provides input upon request by FDA on certain regulatory actions, most commonly decisions on licensure of new vaccines, as well as on more general topics of critical interest to advancing regulatory science. VRBPAC will sometimes, but not always, vote on recommendations. And these recommendations are non-binding, but usually they are followed by FDA in our regulatory decision-making. So the VRBPAC met on October 22nd, last Thursday, for a general discussion of the development, authorization, and/or licensure of vaccines to prevent COVID-19. This was an open meeting with a live webcast accessible to the public and an opportunity for public comment. At this general discussion, there was no discussion of specific COVID-19 vaccine candidates, and also there was no vote on recommendations. Next slide, please. What I have here is the agenda. Following an introduction by FDA, we heard from CDC about the epidemiology, virology, and clinical features of COVID-19, NIH activities in the development of vaccines against COVID-19, BARDA activities in the development of vaccines against COVID-19. We then heard talks from CDC and FDA that you will hear more about this afternoon regarding plans for safety and effectiveness, monitoring following authorization or licensure of a COVID-19 vaccine. We heard about operational aspects of COVID-19 vaccine distribution and tracking from CDC. We heard a talk from the Reagan-Udall Foundation about COVID-19 vaccine confidence. And then the last two presentations were by myself and Jerry Weir at FDA concerning licensure and emergency use authorization of vaccines to prevent COVID-19, considerations related to manufacturing and clinical data. We then had an open public hearing and finally a committee discussion and recommendations, although again, there was no vote on recommendations. Next slide, please. So I've been asked to focus my talk this morning on the FDA presentations. These presentations describe considerations for manufacturing and clinical information needed to support licensure or emergency use authorization of COVID-19 vaccines, as described in recent FDA guidance. Those guidances are listed on the slide. One released in June that covered development and licensure, and then one released earlier this month that was specifically devoted to emergency use authorization. The FDA presentations expanded on and explained the reasoning behind the considerations outlined in the guidance documents, with the goal of providing reassurance that FDA will rely on sound science, established regulatory standards, and a transparent process for evaluating COVID-19 vaccine candidates. Next slide, please. I'm going to focus the remainder of this talk specifically on the clinical considerations because these were of greatest interest and we got the most questions about these clinical considerations during the committee discussion. So what I presented in my talk on clinical considerations was that an EUA, emergency use authorization for a COVID-19 vaccine, may be requested to allow for the vaccine's rapid and widespread deployment for administration to millions of individuals, including healthy people, following a planned interim analysis or perhaps a final analysis in an ongoing phase 3 trial. And in this scenario, a favorable benefit risk determination to support issuance of an EUA would require, in addition to adequate manufacturing information, which I'm not going to cover in detail here, a number of expectations for the clinical data. So first of all, we would expect efficacy data showing protection against SARS-CoV-2 infection or disease with a point estimate of at least 50% versus a placebo comparator and an appropriately alpha-adjusted confidence interval lower bound of greater than 30%. We would also expect that at least half of phase 3 study subjects included in the analysis submitted in an EUA request be followed for both safety and efficacy for at least 2 months following completion of the full vaccination regimen. So a median follow-up duration of at least 2 months after completing vaccination. Thirdly, we would expect safety data from throughout clinical development that includes well over 3,000 phase 3 vaccine recipients to evaluate reactogenicity, serious adverse events, and adverse events of special interest. And finally, we would expect sufficient cases of severe COVID-19 to assess for signals of enhanced disease and also to provide some preliminary information about efficacy against severe disease. Next slide, please. I explained in some more detail the reasons for arriving at this expectation for a median follow-up of at least 2 months after completion of the full vaccination regimen. This follow-up duration, first of all, allows time for potential immune-mediated adverse reactions to be evaluated, given that uncommon but clinically significant immune-mediated adverse reactions to preventative vaccines generally have an onset within 6 weeks following vaccination. This median follow-up of 2 months would also ensure that vaccine efficacy is assessed during the time period when adaptive and/or memory immune responses rather than innate responses are mediating protection. And finally, we consider that this follow-up period would allow for early assessment of waning protection and signals of enhanced disease. Next slide, please. We also made a point that following a successful efficacy analysis that supports issuance of an EUA, further evaluation of a COVID-19 vaccine would be needed for several reasons. First of all, for ongoing benefit risk assessments to consider continuation of the EUA, and then also to accrue additional data to support either licensure and or to inform labeling of a licensed vaccine. And this continued evaluation would include longer-term follow-up for safety, including in larger numbers of vaccine recipients than in clinical trials, as well as in populations with lower representation in clinical trials. The continued evaluation would include or allow for more precise estimation of vaccine effectiveness, more robust assessment of effectiveness against specific aspects of SARS-CoV-2 infection or disease, better characterization of duration of protection, investigation of immune biomarkers that might predict protection, and finally, but certainly not least important, ongoing monitoring for signals of enhanced disease. Next slide, please. Because of this need for continued evaluation, issuance of an EUA for a COVID-19 vaccine would be contingent upon the ability to conduct further vaccine evaluation through a combination of several approaches that include active follow-up of vaccine recipients under the EUA. And in fact, in our guidance, we state an expectation that any EUA request should include a plan for such active follow-up. Also, passive monitoring for clinically significant adverse reactions using established reporting mechanisms such as VAERS. Observational studies, including those that leverage healthcare claims databases. And finally, but again, not least important, continuation of blinded, placebo-controlled follow-up and ongoing clinical trials for as long as is feasible, as well as strategies to handle loss of follow-up. Now, we recognize that it will not be feasible to continue blinded, placebo-controlled follow-up indefinitely, especially in the face of compelling evidence about vaccine efficacy. However, FDA does not consider issuance of an EUA for a COVID-19 vaccine in and of itself to necessitate immediate unblinding of ongoing clinical trials or offering vaccine to all placebo recipients. This is especially true for study participants who may not be included in populations authorized for use under an EUA or included in populations prioritized for use in the case of a vaccine with limited supply. We would also like to make sure that everyone understands that trial participants may choose to withdraw from follow-up for any reason at any time, including to receive vaccine made available under EUA. And so that makes it all the more important that ongoing clinical trials include strategies to handle loss of follow-up should it occur. Next slide. These were the the questions put forth for VRBPAC discussion at the end of the day. We asked the VRBPAC to discuss FDA's approach to safety and effectiveness data as outlined in the respective guidance documents. We asked the VRBPAC to discuss strategies for continuation of blinded phase 3 clinical trials if an EUA has been issued for an investigational COVID-19 vaccine. And we asked the VRBPAC to discuss studies following licensure and or issuance of an EUA to further evaluate safety, effectiveness, and immune markers of protection, and or to evaluate the safety and effectiveness in specific populations. Next slide, please. So I'll spend the last part of this talk summarizing the VRBPAC discussion. First of all, the VRBPAC expressed concerns about public vaccine confidence, consistent with the concerns described in the Reagan-Udall Foundation presentation. It is abundantly clear that there is hesitancy around acceptance and use of COVID-19 vaccines, and that this hesitancy will continue to be driven by perceptions about the speed of vaccine development and about uncertainty and limitations of data that might support vaccine deployment. Therefore, any issues with COVID-19 vaccine deployment could adversely impact public confidence, not only in COVID-19 vaccines, but in vaccines in general. And because of this concern, it is imperative that regulatory actions to make COVID-19 vaccines widely available need to be transparent, effectively communicated, and above all, supported by adequate data. Next slide, please. What we heard from the VRBPAC around data to support an emergency use authorization was, first of all, broad agreement that these data should not be less than the standards outlined in our October 2020 guidance document. Now, there were VRBPAC members who expressed concerns that the standards we outlined were not sufficient. Some VRBPAC members expressed concerns that a median follow-up of 2 months after completion of the vaccination regimen would not be sufficient to support an EUA for rapid and widespread deployment, in particular for vaccines manufactured using novel platforms. Others raised a concern that a successful interim efficacy analysis, especially an early one with more limited COVID-19 cases and wider confidence intervals compared to a final analysis, would not be sufficient to support an EUA for rapid and widespread deployment. Other VRBPAC members, though, commented that 2 months median follow-up could be sufficient to support issuance of an EUA, and that evaluation for rare adverse events and waning protection could best be accomplished by surveillance during use under EUA. Next slide, please. Some VRBPAC members expressed concern about COVID-19 of any severity having been selected as the primary efficacy endpoint in current phase 3 trials. And this concern was related to a concern that analyses to support licensure or emergency use authorization may provide limited information on severe disease, which is where we're really hoping that a COVID-19 vaccine could impact the pandemic. In counterpoint, FDA and some VRBPAC members discussed that the primary endpoints that were chosen for phase 3 trials were selected based on considerations of feasibility, as well as prior experience with preventive vaccines. And to this point, vaccines are typically approved based on data showing prevention of laboratory confirmed disease, regardless of severity. We have a number of lines of evidence and experience supporting that vaccine effectiveness increases with more specific case definitions, including for more severe disease. And finally, analyses to support emergency use authorization will include some information on severe disease, but insisting on adequately powered analyses of severe disease, which will occur with lower incidence and less severe disease, could delay availability of an impactful vaccine. Next slide, please. VRBPAC members also expressed a concern about clinical trial recruitment of and accrual of data in populations most affected by COVID-19, including racial and ethnic minorities, elderly individuals, and individuals with medical comorbidities. FDA discussed that published guidance and advice to COVID-19 vaccine manufacturers has advocated for inclusion of these populations in clinical trials. We do not have a regulatory mechanism for mandating representation of particular populations in clinical trial recruitment. However, as with all vaccine licensures and similar regulatory actions in the past, demographic and medical history data from trial participants will be considered in regulatory decisions around licensure or emergency use authorization of COVID-19 vaccines. For example, considering which age groups should be approved or authorized for use. And furthermore, demographic and medical history data will be reflected in vaccine labeling to inform healthcare providers and vaccine recipients. Finally, as Dr. Bell covered in her introduction, vaccine manufacturers have been publicizing enrollment demographics as well as medical history information for their ongoing trials. Next slide, please. There was some discussion, although not the main focus of the VRBPAC, around considerations for pediatric development and data to support use in pediatric populations. There was clearly some discomfort and concern voiced for the need for safety assessments that include careful evaluation for immune-mediated reactions to vaccines or enhanced disease, including evaluation of cases of multisystem inflammatory syndrome to support benefit risk considerations for both pediatric enrollment in clinical trials as well as for vaccine authorization or approval in pediatric age groups once data on safety and effectiveness in pediatric populations becomes available to even consider this type of authorization or approval. There was some discussion on immunobridging approaches to infer vaccine effectiveness in pediatric populations, and these approaches will certainly benefit from evolving understanding of natural and vaccine-elicited immunity. There was also in the VRBPAC discussion a broad agreement that blinded placebo-controlled follow-up in ongoing clinical trials should continue for as long as is feasible, including after an emergency use authorization. The VRBPAC clearly expressed a concern that if a COVID-19 vaccine were widely deployed under an EUA based on an early interim analysis with data that was more limited than what would be submitted with a licensure application, that this could potentially harm further accrual of critical data from placebo-controlled follow-up. And this concern was based in part on a public statement from at least one vaccine manufacturer of an intention expressly to unblind a clinical trial following an early interim analysis that if it were to be successful. The committee agreed that there was a need for robust strategies for vaccine evaluation following licensure or emergency use authorization to complement placebo-controlled follow-up, and even more importantly, to replace placebo-controlled follow-up once doing so becomes infeasible. Next slide, please. Finally, there was a question raised about expanded access as an alternative to emergency use authorization. FDA explained that expanded access is another regulatory mechanism separate from emergency use authorization for making investigational products available to address serious diseases outside of clinical trials. For a little more information about expanded access, an expanded access treatment protocol could be considered to allow for deployment of a COVID-19 vaccine. The benefit risk considerations would be similar to those for an EUA and would be based on available data as well as the proposed use, including the populations and numbers of individuals to be vaccinated under such a protocol. However, there are other considerations, including considerations that would be important for planning and implementation that would be different from emergency use authorization. And this includes that expanded access treatment protocol would be conducted under IND, investigational and new drug regulations, with requirements that include, but are not limited to, informed consent for clinical investigations, institutional review board oversight, and investigator responsibilities for vaccine providers. Next slide, please. And this is actually the final slide. So next steps, FDA will consider the October 22nd VRBPAC feedback in our efforts to balance the public health goal of safe and effective vaccines to address the COVID-19 pandemic with the obligation to ensure that authorization or approval of any COVID-19 vaccine complies with regulatory requirements for sufficient safety, effectiveness, and manufacturing information to support favorable benefit risk for vaccine recipients. The VRBPAC will be reconvened prior to any FDA action to approve or issue an EUA for a COVID-19 vaccine. And during this or these future VRBPAC sessions, the VRBPAC will evaluate and discuss data submitted in support of the licensure application or the EUA request, and will vote on recommendations as to whether the data discussed support vaccine licensure or support the proposed use of the vaccine under emergency use authorization. That is the end of my talk. Thank you very much.
Thank you very much, Dr. Fink, for that excellent summary of the VRBPAC meeting. We will now take questions from the voting members as well as liaisons. Let me begin by asking this. As mentioned, study participants can withdraw at any time from a study, and certainly with the advent of a vaccine that is EUA approved or fully approved, they have that option to do so. What is the stance of the FDA, or what are the considerations for providing those individuals that withdraw from a study, unblinding them with regard to whether they receive placebo or vaccine?
Right. So FDA cannot mandate to any vaccine manufacturer conducting a clinical trial. We cannot mandate that they unblind or not unblind the clinical trial. Or that they unblind or not unblind any individual participant. It is a matter of ethical consideration as well as scientific consideration, and we would like the vaccine manufacturers who are conducting trials to think very carefully about balancing the ethical and scientific implications of a decision to unblind.
Thank you for that answer. Dr. Alt.
I have two quick questions. Pardon me. One is there a video archive of that meeting we could go back and look at? The ACIP has YouTube videos, for example. So that's one question. The other question is for the expanded access, has there ever been one of this order of magnitude with millions of doses of vaccine? I'm sure several of the members and people listening in have been involved in IRB discussions before, and those are usually small scale in my experience. So I'd be curious to know if there's any experience at the FDA doing expanded access on the scale that we're talking about.
Right. So to answer your first question, I believe there is a YouTube video recording of the VRBPAC available. I don't have the link handy right now, but I will see if I can provide it for dissemination to ACIP members. To answer the second question, we do have recent examples of expanded access treatment protocols that have been used to provide vaccine to thousands of individuals, or tens of thousands of individuals. One example was to address meningococcal B disease outbreak on several college campuses. That was prior to FDA licensure of two meningococcal group B vaccines. And then more recently, there's been an expanded access protocol for use of non-US licensed yellow fever vaccine, Stamrel, to address a shortage of US licensed yellow fever vaccine, YFVAX, in travel clinics across the US. Having said that, we're talking about thousands and tens of thousands of vaccine recipients, not millions of vaccine recipients. And of course, the IND requirements for an expanded access treatment protocol certainly would add some complexities and feasibility concerns to using that regulatory mechanism for widespread deployment.
Dr. Fry.
Thank you. I had a question about safety and vaccine hesitancy. We hear a lot about vaccine hesitancy. The major driving factor for that is safety. And we've had two very nice examples of pause rules or halting rules, if you will, in two of the studies that have occurred already. And this is what we actually like to see. I know it's perhaps novel to the general communities or populations because they're not used to seeing that. But this is a good thing. When people pause a study or hold a study, from further enrollment until these discussions are had and people are comfortable with the decision making. And I was wondering, do all these studies that are being initiated have the same rigorous standard of safety? I know there's FDA guidelines for a certain level of safety oversight, but do you have a sense that all the studies are being scrutinized with the same rigorous standards?
So in a short answer, yes. All of the studies, the phase 3 studies that are currently ongoing have very close oversight from an independent data monitoring committee that regularly reviews safety data and also meets on an ad hoc basis to review and discuss and consider whether any change in trial conduct is warranted following a safety signal, such as a serious adverse event for which causal relationship to the study vaccine cannot be excluded. Furthermore, some studies, in addition to DSMB oversight, and this is more common in smaller phase 1 and phase 2 studies, have pre-specified study pausing rules that are triggered based on certain safety events that might occur.
Ms. McNally.
Thank you, Dr. Fink. This presentation has been very helpful. I just have a question regarding the expanded access. So for those who might not be familiar with the FDA approval processes, can you just provide some additional background on expanded access and how it differs from the traditional FDA approval process and an EUA?
Okay, so first of all, expanded access is not an approval process. The only approval process is licensure. There are several regulatory mechanisms for making investigational products, including investigational vaccines, available outside of clinical investigations, clinical trials. So emergency use authorization is one of those mechanisms, and it is dependent upon declaration of a public health emergency, which is the situation that we are in currently with COVID-19. Expanded access does not require a declaration of a public health emergency, but it does have additional requirements for use of the investigational product as compared with emergency use authorization. And those were the requirements, some of which I listed during my presentation. Expanded access is conducted under investigational new drug regulations, and expanded access use can be of a size that can range from just one patient to hundreds to thousands of patients, depending on the need and the data available to support the proposed use.
So just as a follow-up, how would someone who would want a COVID vaccine then be eligible to receive it under expanded access? And just one more follow-up question is, do you know if the Reagan-Udall Foundation had, during its listening sessions, any discussions with consumers regarding that expanded access?
So if one were to contemplate expanded access use of a COVID-19 vaccine, and I want to stress that this is an idea that was raised, a question that was asked to us by the VRBPAC, and we're still in early stages of considering whether this would be an appropriate mechanism for deployment of a COVID vaccine at all. But if that were to be considered, then the vaccine manufacturer that has an IND an active IND on file with FDA, would submit a protocol for use of the vaccine under expanded access regulations, and would work potentially with other government agencies, as was the case with our yellow fever vaccine expanded access experience, to organize and implement the expanded access protocol. In terms of the Reagan-Udall Foundation, again, this was a question that was raised toward the end of the VRBPAC meeting, and so no, I don't believe that that expanded access was addressed by the Reagan-Udall Foundation's efforts. Over.
Thank you very much.
Not to stifle discussion, but rather to move forward. We will take 3 more questions, the 3 hands that are up at this time, and then we'll move on to other discussions. So I'm sorry if you were holding back from asking a question. We'll try to get to it later. Dr. Lee, please.
Thank you. Dr. Fink, thank you for the excellent presentation, and really appreciate your engagement of VRBPAC and also just all of the work that the FDA has done to get us to this place. I had 2 questions. One was, you had mentioned on one of your slides that there will be, if an EUA occurs, then there would be continued evaluation of COVID-19 vaccine data under an EUA. So my first question is, how often do you anticipate those evaluations would occur? And have there been discussions around potential thresholds that would change a decision as data continue to accumulate? The second question I'll just put out there is, you also mentioned that an EUA would be contingent upon follow-up on safety and effectiveness, and specifically observational studies that leverage healthcare claims databases. So my second question relates to whether or not there have been plans either to use Sentinel as one of the options for that versus manufacturer-led studies, and where those discussions are. Thank you.
Sure. So maybe to answer both of your questions in one fell swoop, the continued evaluation of a vaccine following deployment under either emergency use authorization or licensure would include assessments that are made at scheduled intervals, as well as continuous assessments that would include passive reporting, as well as mechanisms such as rapid cycle analysis. You'll hear about some of these mechanisms later today in the presentations that will be given by CDC and FDA on plans for post-licensure or post-authorization surveillance. So I don't want to step on their toes. I'm certainly not the right person to be speaking with expertise on how those plans will be designed. I do want to make sure that or clarify one thing, that observational studies, including those that leverage healthcare claims data, would not necessarily in and of themselves be a requirement for EUA, but we do see those as a very important part of continued follow-up during deployment. And you'll hear, I believe, in the FDA and maybe CDC presentations about studies that are being planned for that.
Dr. Bernstein.
I want to echo thank you, Dr. Fink. I certainly appreciate your concise and detailed summary of VRBPAC's ongoing extraordinary and challenging work. I just had a quick question of clarification. When you talked about the clinical considerations for EUA, and you had a separate slide of why 2 months was chosen, you mentioned that the median follow-up time period was two months. Was that the median, or is it at least 2 months for all subjects?
It's a median, so at least half of subjects with at least 2 months of follow-up.
What's the shortest period then?
Right. So what I didn't mention in my summary slides, but was included in the more detailed presentation that we gave at VRBPAC, is that we expect that a high proportion of enrolled subjects will have at least 1 month of follow-up. We do recognize that trials will continue to enroll. There will be some lag, and so it will not really be feasible to demand for all subjects to have a certain amount of follow-up to enable a regulatory action to allow for deployment of the vaccine, especially in the face of a very convincing efficacy data. I would also like to note that the phase 3 trials that are currently underway enroll a very large number of subjects very rapidly. And so we do not expect there to be any issue with the numbers of or the proportion of subjects who have safety follow-up that would meet our expectations.
Dr. Sanchit? Oh, I'm sorry, Dr. Bernstein, did I cut you off? Did you have another question?
No, thank you, Dr. Fink.
Thank you. This is Pablo Sanchez. No, thank you for the excellent presentation, really. And 2 questions, I guess a question and a comment. The question is, how soon after VRBPAC makes let's say VRBPAC makes a recommendation that for approval of the vaccine, what is the timeline between the VRBPAC recommendation and the FDA approval, first of all? And then just a clarification, expanded access would be more if a vaccine is studied, let's say, for instance, the Moderna one, which is 18 years of age and greater. Expanded access would be to other populations, i.e. pediatrics, because it was not licensed under that indication. Just, I guess those are my 2 questions.
So let me answer your second question first. So, in order for a vaccine to be made available under expanded access to a large number of recipients, there needs to be sufficient data to support favorable benefit risk determination in the population of those recipients. So I would not envision a scenario in which a vaccine would be made available to pediatric populations outside of anything other than a clinical trial without data to support favorable benefit risk in that population, in those pediatric populations. And that would include safety data and immunogenicity data, at least, to support effectiveness specifically in pediatric populations of the age groups that would be under consideration. So no, expanded access would not be a mechanism for offering vaccine to pediatric populations outside of a clinical trial in the absence of data in pediatric populations. So I hope I made that clear. To answer your first question about timing, we do expect emergency use authorization review and also review of licensure application to be fairly expedited to address the needs of the ongoing pandemic. And so I can't give you a definitive answer about how many days or weeks there might be between a VRBPAC meeting and a vote that includes recommendations to authorize or approve the vaccine, and when that that vaccine actually is made available through FDA action, but only to say that it will be done expediently and as quickly as possible.
Thank you.
Again, thank you, Dr. Fink, for that presentation and answering questions. We'll move on now to Dr. Dubovsky from Novavax, who will present information on their vaccine candidate, NVX-CoV2373. Dr. Dubovsky.
Thank you. I'm pleased to be able to introduce you to our vaccine candidate. Next slide, please. So here's our safe harbor statement. Next slide. So today I'm going to introduce you to our vaccine design. I'm going to describe a single human primate challenge study, and then I'll talk about our clinical development plan as well as data from our phase 1 and phase 2 studies, and a little bit about our plans for phase 3. Next slide, please. So here's our vaccine design. This is a baculovirus expressed recombinant protein. It's a full-length spike, including the transmembrane domain. It includes the usual 3q2p mutations. It self-assembles into a native conformation trimer, and it's further processed into a stable nanoparticle. Our adjuvant is a saponin-based adjuvant. That's a natural product from the bark of a tree. It's purified and processed into these cage-like structures. The antigen and adjuvant are co-administered into a vial where it's a ready-to-use liquid that's stable at 2 to 8 degrees. Next, please. Here's a rhesus protection study I want to talk about. This is a study that was sponsored by Warp Speed and was conducted at the Texas Biomedical Research Institute. We vaccinated these rhesus macaques at day 0 and 21 and challenged them with 10 to the 6 wild-type virus at day 38. On the left-hand panel is the lower airway protection data. You can see in the placebo group on day 2 and day 4, we were able to detect subgenomic RNA, which indicates viral replication was ongoing. On the right-hand side, you can see for both dose levels, and these are the dose levels we've taken into clinical study. You can see that no RNA was detected, indicating that we weren't able to detect replication. On the right-hand side, you see a similar setup, but this is in the upper airway with nasal swabs. You can see in the placebo group, we saw replication on day 2, 4, and 7. And in the vaccine groups, we were unable to detect replication. So this is consistent with the other data we've gathered in other primate studies, as well as in small animals. So next slide. So now I'm going to talk through the clinical development plan. The first in-human phase 1 study was conducted in Australia with 131 subjects, 18 to 59 years of age. And this was a study that was sponsored by ACIP. Next slide. Based on the results from this study, we launched 3 studies, a phase 2 study in the US and Australia, which expanded the age range up to greater than 60, up to 84 years of age. This was also a CEPI-sponsored study. At the same time, we launched a phase 2A/2B efficacy study in South Africa. We've recently expanded this to 4,400 subjects. It includes a small medically stable HIV-positive cohort. This is a Bill and Melinda Gates supported study. And recently we've started a 15,000 subject phase 3 study in the UK. This is supported by the UK Vaccine Task Force and the NIHR. This also has a small sub-study looking at inactivated influenza vaccine co-administration to ensure there are no safety or immunogenicity issues with that. Next. Based on confirmation of the dose as well as safety from the phase 2 study, we're launching a large phase 3 study in US and Mexico with over 30,000 individuals. Next click. I want to spend the rest of the time focusing on the studies I've outlined. So next. This is the phase 1 study design. This has been published back in September. The study had 131 subjects, aged 18 to 59, and there were 5 dose groups. There was a placebo group. There was a dose group that had 25 micrograms of antigen with no adjuvant given twice. There was a high dose antigen adjuvant dose. And there was a group that had 2 doses of 5 and then 2 doses of 25 micrograms given twice. The study is fully enrolled, but follow-up is ongoing, and we're blinded to the individual vaccine and placebo allocation. The day 35 data, which was the basis of the publication, has been reviewed by our independent safety monitoring committee and submitted to the FDA in advance of the next study. Give me a click, please. I'm going to be focusing much of the attention on these groups, the placebo group compared to the two active two-dose groups. Next slide, please. So from a safety summary perspective, there were no serious adverse events. There were no AESIs. From a treatment emergent adverse event perspective, all of them were either mild or moderate, and they were balanced in the active arms. There were no severe events. And from a reactogenicity perspective, the majority of all subjects reported either no or mild reactose symptoms. And the mean duration of these were less than 2 days. I'm going to review these in the next two slides. So next, please. What's displayed here are the local reactogenicity symptoms collected 7 days after each dose. Along the top swim lane is after vaccination 1, and the bottom swim lane are after vaccination number 2. On the left-hand side, I've pulled all of them together so you can see kind of an aggregated local reactogenicity display of data. And you can see that the majority of symptoms were either no or mild symptoms. Pain and tenderness were most commonly reported, and the mean duration was less than 2 days for each of these events. Next, please. This is similar data, but for systemic reactogenicity symptoms. Once again, the aggregated data is on the left-hand side. You can see that there were more symptoms after dose 2 versus dose 1, and that was true in the placebo group as well as in the active groups. The most common symptoms that were reported were headache, fatigue, and myalgia. And once again, the mean duration was less than 2 days. Next, please. So let's move on to immunogenicity. Here, what's displayed is our anti-spike IgG ELISA kinetics. And we can start with the placebo group on the bottom in blue. You can see after 2 doses delivered on day 0 and 21, there was pretty much no response. After the 2 doses with no adjuvant, you can see we got a bump after dose 2 that was stable throughout the observation period. In purple, you can see the single dose of adjuvanted material. You can see we got a nice response after dose 1, which stabilized. And finally, you can see with our 2 doses of interest, we got a nice bump after dose 2 with remaining high levels together. So from this data, we conclude that adjuvanted vaccine is better than non-adjuvanted vaccine. And furthermore, the 2 doses is superior to a single dose. You can see there's a large dose-sparing effect with the adjuvant because the 5 microgram dose group is really comparable to the 25 microgram dose group. Next slide, please. So what I've displayed here are the peak immune responses on day 35. On the left-hand panel, I have IgG. In the right-hand panel, I have the wild-type neutralization responses. Just to set the assays, the NEUTs were conducted by the Dr. Fryman lab at the University of Maryland School of Medicine. And these are 100% wild-type neutralization responses. So maybe give me a click. Thank you. So let's focus on the left-hand side for the IgG responses. And you can see that the 2 dose groups with adjuvant performed quite well. In gray in both panels is a panel of convalescent serum that was donated by Dr. Pietro from the Baylor College of Medicine. And this is an uncurated panel of serum that ends the same samples on both the IgG and the neutralization responses. So what you can see here is that we achieved 100% seroconversion for IgG as well as for microneuts. And furthermore, in a post-hoc analysis, if you look at the means, you can see that for IgG, we achieved a 5 to 7-fold higher than the convalescent serum. And for the microneuts, 3 to 4-fold that was reported for the convalescents. And additionally, the confidence intervals do not overlap. So can I have the next slide, please? We were interested in understanding the quality of our immune response. So we looked at a correlation between the neutralization response and the IgG response. On the left-hand panel, you can see the Baylor panel, and you can see that there's a nice correlation between IgG and NEUTs, Pearson correlation of 0.96, it looks like. So this indicates that when people get sick with wild type infection, they generate antibodies, and those antibodies are capable of neutralizing the virus. In the middle panel, I have data displayed that is from our 25 microgram dose group with no adjuvant. And you can see all the correlation breaks down. Although we can generate antibodies, not all of those antibodies can neutralize. On the right-hand panel, I've displayed the adjuvanted dose groups. And you can see that we recapitulate the pattern seen with wild type seroconversion, and our correlation becomes tight again. So to us, this indicated that the matrix was important in generating the kinds of immune responses we wanted, that we generate a functional immune response against a broad range of antibody titer. From a safety perspective, we think this is important because some people have postulated that antibody that does not neutralize may lead to some enhanced illness and sickness in the future. Next slide. So now I'm going to turn to the cellular immune response. What I've displayed here is placebo, the 2-dose 5 microgram dose group, and the 2-dose 25 microgram dose group. And under each grouping, I have 2 panels, one that shows Th1 cytokines and one that shows Th2 cytokines. And you can see in the placebo group, pretty much the signal was flat. However, in both the 5 and 25 microgram dose group, you can see we detected a signal for IL-2, interferon gamma, and TNF-alpha. On the Th2 cytokines, we probed for IL-5 and IL-13, and you can see a small increase in those cytokines. And this really recapitulates what we've seen in small animal models, indicating that this adjuvanted platform are capable of generating a Th1 bias immune response. And this is consistent with our previous evaluation of this platform in people. Can I have the next slide, please? This is an alternate display of the data where we were looking at polyfunctional CD4 cells. And you can see for both dose groups, we have a relatively large proportion of CD4 cells that generate double cytokines as well as triple cytokines, especially when that's compared to the double cytokine for the Th2 analysis. This is the gating strategy here was to look for CD45 CCR7 negative CD4 cells. So these are thought to be effector memory cells. So we postulated this is going to help with our memory response going into the future. Can I have the next slide, please? So from our phase 1 conclusions, the safety and reactogenicity profile was reassuring for both dosage levels. From the immunogenicity conclusions, the matrix was required to induce optimal functional immune response. 2 doses of vaccine administered 21 days apart were superior to a single dose, and the 5 and 25 microgram dose group generated a comparable immune response. And we induced a Th1 bias immune response with high levels of neutralizing antibody. So both dose levels were acceptable to take into the clinic, and the regulatory authorities, as well as our independent safety monitoring committee, agreed with this conclusion. Next slide. What I have here is the design of our phase 2 study. This is a study that's fully enrolled, but is ongoing now, and we are blinded to individual allocations. Let me just walk through the groups. We had a placebo group. We had a 2-dose group of 5 micrograms. We had a single 5 microgram dose group, 2 doses of 25, and a single 25 microgram dose group. And once again, I'm going to focus on the 2-dose groups because they're most relevant to the way we plan to use this vaccine in the future. The reactogenicity data I'm about to show you has been reviewed by our independent safety monitoring committee, and some of it has been submitted to the FDA in advance of our phase 3 study. But I have to say this is live data, so in a subpopulation of the total vaccinees. So can I have the next slide, please? So this data display is very similar to what I showed earlier. On the left-hand side is an aggregate of all the local reactogenicity events in all subjects. And I've broken it down by age group on the right-hand side of the panel. And you can see that pain and tenderness were once again reported most frequently. And we had increased rates in the adjuvanted groups, especially after dose 2. The reactogenicity events were attenuated in older adults. This is exactly what we expected from our previous experience. And the aggregated terms are the same as I showed previously. They were pain, tenderness, erythema, and swelling. Next slide. This is a companion slide to the previous one, but it looks at systemic reactogenicity events. Headache, fatigue, and myalgia were most frequently reported. And we saw increased rates in the adjuvanted groups, especially after dose 2. And once again, we saw attenuation of these signals in adults greater than 60 years of age. Next slide. So now I'm going to touch a bit on our planned US-Mexico phase 3 design. So this is a phase 3 randomized observer blinded placebo-controlled study. It's really very similar to the other studies you've heard about because of the harmonization efforts put in by Operation Warp Speed. Ours, however, is randomized 2-to-1 to receive 5 micrograms of antigen plus matrix versus placebo. We're giving 2 doses, 21 days apart. Anticipating up to 30,000 adults. The protocol specifies that we want to focus on at least 25% adults greater than 65 years of age, 25% those with high comorbidities, and various racial breakdowns is listed here. This is an endpoint-driven study, as with all these efficacy evaluations, and we plan to do an interim at 72 and 108 endpoints, and a final at 144 cases. The primary endpoints are very similar to the other studies. It's the prevention of PCR-confirmed mild, moderate, or severe COVID illness occurring 7 days after dose 2 in baseline seronegative adults. And the follow-up, as with all these studies, is through 2 years. Next, please. So in summary, the vaccine is based on Novavax's baculovirus nanoparticle platform technology. The safety database for the platform includes over 12,000 vaccinees, and the safety database includes over 12,500 vaccinees that have been adjuvanted with our Matrix 1 adjuvant. We're planning to use 10-dose vials with transportation and storage conditions at 2 to 8 degrees. This is a preservative-free preparation, and no admixing or reconstitution is required. We're planning on administering 0.5 ml intramuscularly, 21 days apart. In our opinion, the preliminary safety profile is reassuring and favorable reactogenicity profile as well. Our peak immune response occurred at 14 days after dose 2. And we have a favorable immunologic profile. We have a robust neutralizing antibody response with a polyfunctional CD4 Th1 biased cellular response. This is a immunologic phenotype that has been postulated to be preferable. And finally, efficacy evaluation is ongoing. I'll conclude there. Thank you very much.
Thank you very much for that excellent presentation. We'll take questions now from the voting members, if there are any. Let me ask 2, if I may. So the first is regarding your convalescent serum panel. Can you provide us with any information as to when that serum was obtained in the patient's illness, how many days after recovery? And the second question is regarding your spike protein. So it can exist in 2 forms. It's metastable. Is the protein locked into either the pre-binding or the post-binding form in your vaccine?
So let me take on the first question first. The details of that convalescent serum were highlighted in the Keach publication, but in general, these were a median of 19 days after diagnosis. A small number were hospitalized, a small number were asymptomatic. The vast majority were those which we would consider having moderate illness. They showed up to the emergency room for care, and that's where they were recruited. The full-length protein has the stabilizing mutations, the 2P mutations, and that locks it into conformation. Greg, do you want to say anything more about that?
I think that's right. It's in the pre-binding form. And just last week, the structure was published in Science as it was described by Script. They did an analysis of the drug substance. So you can reference that in Script's, in Science, and you'll look for the NVX2373.
Thank you for those answers. Dr. Fry.
Thank you. Thank you for this very nice presentation. I was curious to know what your thinking was about the mechanism that would cause the adverse events or local and systemic events post-vaccination to increase after second vaccination. And are you giving the both vaccines in the same arm, and do you think that might play a role? Thank you.
Thank you for the question. We don't mandate, but we recommend that alternate arms be used, and that just so we can better follow the local events. It's very typical for the reactogenicity to increase after dose 2, and that's because, as you saw, the immune response also bumps after dose 2, and these local reactogenicity events are thought really to be related to the immune response and the local immune response that's generated with vaccination. It's consistent with what we've seen previously and consistent with what we saw in our phase 3 influenza program.
And do you think that might be due to the matrix and adjuvant?
Oh, yeah. So yes, I didn't show the data, but the data is available in the publication. The unadjuvanted groups had a lower level of reactogenicity.
Dr. Szilagyi.
Yes, thank you for a nice presentation. Just a very quick question about the phase 3 planned trial. Do you have targets for what proportion of the 30,000 adults would be in the United States?
Yeah, currently the plan is that a very large portion indeed will be in the United States. Lisa, do you have some estimates of that? Maybe not.
Yeah.
Yeah.
Can you hear me?
Okay, so go ahead. Yep, you're on.
Yes, we estimate that roughly 10% of the population will be in Mexico and the rest in the US.
Okay, thank you.
Dr. Lee.
Thank you for the excellent presentation. It certainly seems very promising. And I will say that with all these vaccines, it does seem like local and systemic reactions are going to be extremely common and should be anticipated regardless of COVID vaccine type. My question relates to one of your slides actually had a grade 4 reaction, and I was wondering if you could comment on what that was.
Yes, this was actually in the placebo group. It was a fever that was reported to be greater than 40 degrees. We think we're blind to the individual, so we can't know, but that's what we've been indicated from our safety monitoring group. We don't believe that saline causes high fevers. In our opinion, this is likely to be a data entry error. Like I said, this is uncleaned live data, and this may resolve as cleaning progresses.
Thank you.
And just based on the reactogenicity profile, this is quite favorable. I don't know if you noticed, but we didn't really have a fever signal in the phase 1 study at all. This is really comparable to other licensed vaccines.
Are there any questions from the liaisons? Dr. Fry, your hand is still up. I just want to make sure I'm not missing you for another question.
Yeah, thanks. Thank you. I have one last question. So if I heard you correctly, you said you were you are doing a phase 4 study currently in Mexico. Does that mean it's, I thought phase 4 were post-licensure studies.
I think the question was around grade 4 reactogenicity events. The phase 3 we plan to do in the US has a small portion of it being conducted in Mexico. Our other 2 studies in the UK is a phase 3 study, which is about halfway enrolled now. And in South Africa, it's also a phase 2A, 2B, and that's also about halfway enrolled.
Okay, thank you.
Dr. Freihofer.
Sandra Freihofer, American Medical Association. On your slide 18 and on your slide 19, you note that the reactogenicity is attenuated in adults age 60 and older. Does that sort of indicate that this vaccine may not work as well in older individuals?
So this is a pattern we've seen and seen with pretty much all vaccines. We know that the immune responses are attenuated in older adults. Immunosenescence is part of what happens in life. The impact on efficacy really isn't unknown, and we haven't really displayed the immunogenicity data because we don't have that available to us yet. So that may be a lead to understand what's going on.
Dr. Whitney Williams, please.
Yes, thank you, Dr. Romero. Thank you for this presentation. I noticed in the phase 2 trials there were, I think, 240 HIV positive patients. Any plans going forward in any of the phase 3 trials to include that group?
Thank you for that question. That's in the South African study, and their HIV rates are extremely high. So part of the discussion with the regulators and the civil society groups there was the need to include those because that's the population which we will be vaccinating in the future. Once we understand the safety and immunogenicity in that population, we'll be better positioned to make our future plans. And those cohorts are being vaccinated right now. So we should have data in the not too distant future.
Thank you very much.
Philip, you may want to note also that the phase 3 trial does not exclude stable HIV infected individuals.
That's right.
Are there any other questions from the voting members or from the liaisons before we close? Seeing none, let me thank you again for this presentation, and we'll move on to the next presentation by Dr. Sadov of Janssen's SARS-CoV-2 vaccine program. Dr. Sadov, please go forward.
Yes, thank you very much for the opportunity to present our program to you. I'm Jerry Sadov, and I'm the lead clinical investigator for the phase 1/2 and the current ongoing phase 3 study. Next slide, please. This is just our forward-looking statement. Next slide, please. So first, I'd like to tell you just a little bit about our technology platform, which is the foundation for our vaccine. We use a replication-incompetent human adenovirus 26, which expresses the target antigen. So we've taken the region out of the virus so that it can't replicate. So it invades the cells and makes the transgene and its own antigens, but it can't really assemble them into a replicating virus. So the antigen is not on the surface of the virus, but only on the surface of the cells that the virus has gone into, and it's membrane bound. And this vaccine in our program has induced very good humoral and cellular immune responses, antibodies response against structural proteins with neutralizing antibody, and also it has unique function. We've noticed that they have unique functional characteristics because they're probably presented in the context of the cell. They have ADCC function, which we've recently shown to be important in malaria challenge studies as a unique feature that's in addition to the neutralizing antibody that we get. On the cellular side, we see CD4 responses with Th1 signature and CD8 T cell responses. We see no sign of vaccine-associated respiratory enhanced disease in preclinical models, and we have a very vigorous RSV program even after breakthrough infection. And extensive clinical experience with our vaccines. We've been into over 110,000 participants that have been vaccinated with the AP26 vector, and we've shown there that they have a favorable safety and tolerability profile in the populations that are studied to date. And just as you know, on July this year, we obtained approval for our Ebola vaccine from the EMA. Next slide, please. Now, we took the approach to look at a number of vaccines, constructs of the S, because of the instability problems that were noted before, and also to look at other theoretical considerations. We've had a lot of experience with our RSV and HIV programs on how to construct stable expression vectors. And so we've looked at stabilization, the signal peptide, expression of the antigen. We also looked at manufacturability, and then taking these different constructs, for about 12 of them, we looked at immunogenicity in preclinical animal models. And based on that, we were able to find one which was clearly superior in terms of its immunogenicity and the other considerations. And it encodes a full-length membrane-bound S protein with stabilizing mutations and a native initial signal sequence. Next slide, please. So this is just one example of this vaccine in the non-human primate challenge model system, where at week 6, compared to controls, which you see all of them developing both messenger RNA copies that we can detect in the bronchoalveolar lavage, as well as the nasal swab. They were all infected early with fairly high levels. And in the vaccinated groups after a single vaccination, we were able to show complete protection in the bronchoalveolar lavage and only one animal with nasal swab. In other data, we've done down dosing studies where we've shown that even when we reduce the vaccine, which does have lower immunogenicity at very low levels, we've still been able to get substantial protection. And this leads us to the belief that the immunogenicity that we get will be protective. Next slide. So based on that, we started a phase 1/2A study to look at the safety, reactogenicity, and immunogenicity of this investigational vaccine. And we conducted it in healthy adults aged 18 to 55, as well as adults 65 years and older. And we used 2 different doses of the vaccine, 5 times 10 to the 10th viral particles, or 1 times 10 to the 11th viral particles. And these were administered either as a 1-dose or a 2-dose regimen. We have the data on the 1-dose. We're just accumulating the data on the 2-dose, so I'll focus mainly on our data after 1 dose. So cohort 1, there's 400 subjects that were 18 to 55, and this is still ongoing. Cohort 3 was greater than 65 years of age, 375 years of age. And then cohort 2, which is about to start in 270, we'll be looking at duration of the immune response and the ability to boost at various times, if necessary, 6, 12, and 18 months, with also looking for amnestic responses. Next slide, please. So this is just the details of that study in the cohort 1 and cohort 3 being identical. You can see we have 2 doses at 5 times 10 to the 10th, separated by 57 days in group 1, and then a single dose. And then group 3 and 4 are duplication of that for that 1 times 10 to the 11th, and then a placebo. And we're going to talk about the data after day 29, which is the interim analysis. And the primary analysis will be occurring within the next few weeks. Next slide, please. So this is the immunologic data that we saw, both ELISA on the left and wild-type VNA on the right. And you can see that we have very good seroconversion rates with our in ELISA at 99% and with the slightly lower dose, and similar 99% with very reasonable GMTs for neutralizing antibody levels. And when we looked at the cohort 3, the adults older than 65 years of age, there we have data at day 15 and 29. We saw that there was very little difference between the elderly adults and the younger adults in terms of immune response, virtually indistinguishable, and that for the ELISA, we saw a rise between day 15 and day 29. But if you look at the wild-type VNA on the right, once again, we had very good responsive rates, 92% at both doses. And in the elderly, we similarly had 90% and 91% with comparable GMTs and overlaps. But here we saw that the GMTs for the neutralizing antibody come up a little quicker than the ELISA, and so we have a very good response at day 14. So therefore, in our phase 3 trial, we've decided that we would start counting cases 2 weeks after the single dose immunization. And from this data, it's clear that we don't have differences in immunogenicity between the younger group and the elderly group in terms of wild-type neutralization or ELISA, and also that the doses seem to be at the flat part of the dose response curve, which allows us to pick the lower dose as a single dose regimen for going forward. Now, the next slide shows some of our cellular data. Next slide, please. And here we looked at CD4 T cells as well. We had a very good response of 76% and 83% for the 2 different doses in the younger age group, and slightly lower response in the older age group. And these are Th1 responses measured by gamma interferon and IL-2. If you look at the CD4 T cells that had Th2 responses to Th2 cytokines, we only had 2 individuals that responded with Th2. The rest were completely negative. And the ratio in those two between the Th1 and Th2 was 28.9 and 20.2. So clearly, it's a Th1 type response that dominated in both the younger adults and the elderly, with practically no Th2 response at all. Next slide, please. Now we also, with this vector, are able to induce in individuals CD8 T cell responses. And these CD8 T cell functional responses that express gamma interferon may play a role in protection as well, although we have yet to prove that. The CD8 responses at 51% and 64% at the in the younger age group, and slightly lower at 36 and 24% in the older age group, do reflect a fairly high percentage of CD8 T cell responses. I would point out that we do have quite a stringent test for discriminating between background and the higher and the responses. And so compared to some other techniques we've used, these GMTs might be a bit lower than we would normally see. Next slide, please. I just wanted to go over the adverse event profile. As was mentioned before, this vaccine does give some systemic adverse events, and this is blinded data between the groups. This is not unblinded data, but there's 402 subjects in the 18 to 55-year-olds, and you can see that of overall participants, there's around 50 to 60% of systemic adverse events, and having any systemic. Mainly we see fatigue, headache, and myalgia. All of this is quite transient and lasted not more than 1 to 2 days at the most, and then completely went away. On the local symptoms, we also had some local symptoms. In terms of pyrexia, we had some fever in the younger age group that was even considered grade 3, 38 to 40 degrees centigrade, 102 or above for Fahrenheit. If we look at, these were also very responsive to antipyretics, and we didn't judge that we needed to use antipyretics prophylactically. In the elderly population, we saw far fewer systemic reactions. Local reactions were similar, and in terms of fever, we didn't see any grade 3 fevers and very low fever rates. So once again, the reactogenicity in the elderly population was much lower than in the younger population, but the immunogenicity seemed to be comparable. Next slide, please. So based on this, as I mentioned, we designed a phase 3 efficacy trial after which was a multi-center randomized double-blind placebo-controlled phase 3 study evaluating efficacy and safety for the prevention of SARS-CoV-2 mediated COVID-19. And our case definition is moderate to severe, although we have secondary endpoints for mild and for combined mild, moderate, and severe, as well as other secondary endpoints for medical utilization. But the primary endpoint is moderate to severe. The study is being conducted in Argentina, Brazil, Chile, Colombia, Mexico, Peru, South Africa, and the United States, with a target of about 50% of the subjects being in the United States. We also, from the beginning, have planned to enroll a diverse population in the United States, and of course, diverse populations in the countries outside the United States. Our study does not have a fixed interim analysis. We use a continuous sequential monitoring technique where we look at the number of cases in the vaccine group versus the total number of cases on the x-axis, and we have lines for safety as well as for efficacy, and continuously monitoring after every case whether we cross a safety boundary or we cross an efficacy boundary. And the efficacy is calculated from the first 20 cases up to a total of 154 cases with the alpha distributed during that time. Now the full protocol is completely accessible, and it's been published, and I can show you you can see the link. So it's being done in healthy adults 18 to age and above, and in 2 cohorts that started simultaneously where we have safety running for both of them. Our estimated enrollment is 60,000, and it's simply a single dose of 5 times 10 to the 10th, add 26, CoV-2S, our vaccine, or placebo. And the endpoints are the number of participants with the first occurrence of molecularly confirmed moderate to severe COVID-19. We would hope to have a planned follow-up of 2 years. We will attempt to follow participants as long as we can, regardless of what happens. But of course, as been discussed earlier, we have plans for how we analytically would deal with dropouts and people that wanted to get the other vaccine, as well as making them knowing when they would, if they had placebo or vaccine. Next slide. Now, I did just, before I close, I just did want to discuss briefly the pause that we had. As has been mentioned before, pauses are not uncommon in these kinds of studies, and we as other companies are dedicated to the safety of our participants. And when we're enrolling large numbers of study patients, participants, you will see pauses. This particular pause was in an individual that had a safety event, and because it was part of our safety run-in, we had automatic stopping rules or pausing rules that occurred when we had this event, and this was judged by the DSMB to be appropriate. So basically, we evaluated this case with the DSMB, with their consultation. We had outside experts in relevant fields come in. We did extensive evaluations of the case, and from a laboratory and other point of view. And it's a complicated case that had multiple potential causes, which have not completely been determined. But the DSMB and our experts and our company safety group has believed that there's no relationship between the vaccination and the event. Our DSMB, furthermore in consultation with the FDA, agreed to restart the trial. And the trial has restarted in the United States, and enrollment is going on, and in some countries in South America, and the applications to the appropriate regulatory authorities and IRBs are going on right now, so that the trial should continue to enroll over the next week or so. Now, we do plan to disclose clinical data for our COVID-19 trials once those data are presented or published at pre-specified milestones, and we'll proactively disclose regulatory trial holds as requested by health authorities. Next slide, please. Now, we are preparing for this vaccine to be deployed widely once we reach a signal and we have approval from the FDA and other regulatory agencies. We plan to put it in a 2-hour glass vial that has 5 doses per vial. And there's the other packaging that you can see. And we have an anticipated storage conditions under the EUA where it would be stored by us in our facilities at -20 for up to 2 years. But at the end user stage, when it's shipped to the end user, right now, it would have 2 to 8 degree storage for up to 3 months. But once we get more data, which is evolving right now, we would hope to have much longer stability, up to 9 months or longer, like for our other vaccines in this platform. And once it's opened, since it's a 5-dose vial, it can be used for up to 6 hours if stored at 2 to 8 degrees centigrade. Next slide. So that ends my presentation, and Thank you very much for your attention.
Thank you very much, Dr. Sadov. We'll open it up to questions. I see Dr. Alt has his hand up first. Dr. Alt.
I wanted to ask the representatives from Novavax if they have any plans about a phase 2 or 3 trial in pregnancy. They just published results of an RSV vaccine that uses the same platform in pregnant women. It was just in the New England Journal of Medicine fairly recently. So I wanted to know if they have plans to use this platform in pregnancy.
Thank you. We're just in discussions with regulatory agencies how to best go forward. The study that's published was not formulated with this matrix adjuvant. So there are other considerations which need to be taken into place. We're concluding our development and reproductive tox study, and we'll make decisions based on that.
Yes, and maybe I could address that as well from Janssen point of view. We are currently doing studies in our Ebola program in pregnant women. And as soon as our toxicology data is available, probably within the next month, we would be planning to start studies in pregnant women because we understand how important it is in this age group to be able to vaccinate in that population.
Thank you.
Dr. Palin.
Hi, thank you very much for this nice presentation. I have 2 questions for you. One is on the adverse event profile. I believe the data you presented had both doses combined. And is there a plan to look at that data by the doses, particularly the 5 to 10 to the 10 that is being used in the clinical trial, phase 3? And then the second question is, would you please expand on the discussion? The vast majority of the CD4 T cells were in the Th1 bias response, and then there was one in each age group that had a ratio of Th1, Th2. Could you expand on that conversation, please? Thank you.
Yes, so on the Th1, Th2, basically we feel that in looking at our platform, using this, that the Th1 bias in the animal models we've looked at in RSV so far, that this may be strongly indicative of not being the type of pattern that you see with enhanced respiratory disease based on vaccine. So that in combination with antibody is a telling signal on that. And if you have a Th2 response with a predominantly Th1 type response in that individual, that seemed to be, in combination with neutralizing antibody, to be the type of immune response that is not associated with enhanced disease. And your first question again? I'm sorry.
No, thank you. The first question was about the adverse events profile. Is there a plan to look at that data by dose?
Right. The FDA and our other regulatory agencies have seen unblinded data. A limited number of individuals have seen unblinded data. And there is, I can just say that there is a trend toward less reactogenicity in the lower dose group. We plan to publish this data after we have the second dose safety data from the 2-dose regimen of the ongoing study, so that we would then publish the safety data in the 2 groups, both in terms of lower and higher dose, so that would become very clear. And that should be available fairly soon, as but we want to maintain the blinding in the phase 1 data and a phase 1 study until we've looked at the safety data in the elderly age group at the second dose.
Dr. Szilagyi.
Yes, thank you for a very nice presentation. I had a question about the phase 3 study. I was interested that the primary outcome is moderate to severe disease. And it looks like healthy adults. So does that mean that adults with diabetes, cardiac disease, lung disease, obesity, et cetera, were excluded, are excluded from the phase 3 trial? And also, can you also let us know what percentage of the enrollees will be in the United States as planned?
Yeah, so the primary endpoint of the trial is moderate to severe disease. We, in our safety run-in, we are excluding comorbidities that are associated with accelerated disease, and that would be in the first 2,000 participants in the younger age group and the first 2,000, and they can have other comorbidities. After those first 4,000 are enrolled in each age group separately, we will enroll individuals with all the comorbidities associated with accelerated disease, and we do enroll, even in the initial ones, all the comorbidities. So specifically, diabetes, hypertension, all of those will be a major part of the trial. Yes, they're all included. So, and the moderate definition was picked primarily because in our experience with, and others, I believe, with the vaccines against respiratory diseases, vaccines have a better chance of working against more severe disease and not necessarily such a high level of protection against mild upper respiratory disease. And so this provides two things for us to go for a moderate to severe endpoint. One, a better chance for the vaccine to show its true effectiveness. And second of all, a more clinically relevant endpoint to some extent. But we are looking very carefully at mild disease, as well as in the trial we've built in at 3, 6, and 12 months bleeds to look for seroconversion against the virus, so that we, and this is an assay that does not include the S protein, so it isn't interfered with by the vaccine, to look for asymptomatic disease. So we should have a very good understanding of the vaccine's effectiveness against asymptomatic and all different forms of the disease. Furthermore, in every case that we find, we are monitoring them very closely by getting nasal swabs every other day and saliva samples until they're two consecutive ones are negative. And then we will compare the control group to the vaccine group to see if the vaccine has an effect on the viral load that is even in the breakthrough cases. Now, we're doing this because in a recent RSV challenge study that we conducted, we showed very good effectiveness of the RSV vaccine in the upper respiratory tract to prevent infection. But in the breakthrough cases, the number of viral particles and live virus in the recipients of the vaccine was dramatically lower than in the non-vaccinated group. So we may have an effect on transmission, even if we can't prevent complete protection in the upper respiratory tract. So therefore, we're measuring that as well.
Great, thank you. And any estimate about the proportion that will be in the United States?
In the United States, we estimate, and we are attempting to have at least 50% of the individuals be in the United States compared to the rest of the countries.
Thank you very much.
Dr. Kimberlin?
Thank you for a very interesting presentation. I commend you and the other manufacturers on your rapid yet deliberative approaches to this unprecedented situation and the need for a vaccine, a safe and effective vaccine. Toward that end, transparency, as you all have done with posting of your phase 3 protocol, is critically important. I'm interested in knowing more about the subject halt for the phase 3 study that was discussed in really very broad terms, and I think we all would benefit by knowing more about what at least system that was related to.
Yeah, thank you for the question. Yeah, we have been very transparent about the case with the regulatory agencies, IRBs, and the investigators have been fully briefed on the case. We have been reluctant to discuss the case in public, however, for the reason that the study is still blinded. And second of all, there is a matter of patient confidentiality, which we are really very concerned about. So at this point, we haven't really felt it appropriate to disclose the nature of the adverse event, other than to mention that it has been fully discussed with independent consultants, the IRB, the IRBs, the IDMC, and the regulatory agencies including the FDA, which have all agreed we should go forward. We will be disclosing as much information as we can in a timely manner when it's appropriate, but fully respecting the patient confidentiality. So that's basically why we haven't given information that might lead to speculation, which is not warranted based on the data that we have.
Dr. Freihofer.
Sandra Freihofer, American Medical Association. 3 small questions. Number 1, will diversity data of your trial participants be posted on the website in any way? And another question, I know you won't disclose. I just hope you understand how important transparency is to developing vaccine confidence. Could you even give us the age of the person who had the safety event? And a third question, you mentioned the Th1 bias T cell response and vaccine enhanced respiratory problems. Are you looking at any of the thrombotic or inflammatory responses in your study?
Thank you for the question. Yeah, so we will be posting a diversity figures just like the other companies will. And yes, we will be very transparent about that. And we do agree with you that transparency is very important for very many reasons, especially under these circumstances. And so we will disclose as much information as possible that does not in any way violate the patient confidentiality. I can tell you that this was a young individual in the younger age group and not in the elderly group. In terms of the thromboembolic events, it's not an adverse event of a special interest, but it's something that we are looking at very closely. We have a electronic device that every single individual in the trial gets, as well as a pulse oximeter and a thermometer, and we have a trigger for symptoms that may be anything related to COVID. And it's a broad trigger so that we can have the participant immediately be taking their oxygenation levels and temperature, even for the mildest cases. And of course, the any thrombotic event is considered a trigger, and any symptom of that is even a trigger to go beyond that, but to immediately call the site or their own doctor, and that's electronically triggered automatically when something similar to a thrombotic event might occur. So we are monitoring that very closely in all the participants in the trial.
Thank you. So we've almost used up our break and we'll be going into the next session, but given the importance of this line of questioning, I'm going to continue to take additional questioners. So Dr. Lee, please go forward.
Thank you. I wanted to ask on the slide that you had showed with regard to the ELISA and the neutralizing antibody assays, I noted that in comparison to the convalescent sera, it looked a little bit lower than convalescent sera. And I'm just wondering, so my question is, do you think that is meaningful in any way? And then the second question I have for the broader group, I think, is around how we standardize convalescent sera and whether or not this is comparable across trials, because I'm assuming these all come from different lots or different, locations.
Yes, I didn't go into the technical details, but on that slide, you might have noticed that there was a dotted line across the top for the antibody, the neutralizing antibody. And that line represented the upper level of quantitation. And so that meant that there were quite a few sera in our both younger and elderly group that could have higher dilution than we hadn't measured yet. And that has been measured. So the geometric mean titers will go up somewhat. But I would say that we're slightly lower than the convalescent sera. I strongly agree with your second point that we need standardization of these sera so that we could directly look from a comparison point of view so we can learn how relevant it is. And so therefore, we would also want to have standardized sera that can be looked at. We've looked at several different panels and have different results from the different panels. One panel were higher, another panel were slightly lower. So we think that this standardization would really be quite useful for the field and would encourage that to occur. As far as the implications are, we think that being near what the convalescent sera is probably adequate. And we don't know right now how difficult this virus is to neutralize, whether we need extremely high titers or low titers. Some viruses like hepatitis A, you can barely measure the neutralizing antibody and you're protected. Others require more. So we have to learn that.
Ms. McNally.
Thank you, Dr. Sadoff. I have a really quick follow-up question based on Drs. Kimberlin and Freihoffer's question. So you mentioned that you would be disclosing in a timely manner the information about the unexplained illness in the study pause. But given the abbreviated timeline that we have, can you be more specific regarding what the timely manner looks like?
I said we would be disclosing the data based on the dose ranging. In other words, what would be the blinded data on doses and versus safety and immunogenicity would be done fairly shortly when we finish the second dose evaluations. In terms of evaluating more information about the pause and the case, that will be determined, like I said, based on confidentiality and unblinding considerations. And it may turn out that we'll be able to disclose more of that in a short period of time, depending on how the case evolves and also whether or not the confidentiality issues may not be an issue at some point. So that's what I can say right now.
Thank you.
Dr. Alt, your hand is up. I just want to make sure that you don't want to follow up with another question.
Nope, that's a mistake.
Thank you very much. Dr. O'Leary, go forward.
Yeah, Sean O'Leary, Pediatric Infectious Diseases Society. I just want to echo the other sentiments. This confidence in these vaccines is so crucially important that transparency takes on extra weight in the current environment. And I think you very well may be justified in these confidentiality concerns, but I think from the perspective of the public, in a trial of thousands of people, that's going to be a hard pill to swallow. So I really encourage you to be as transparent as possible.
Well, yes, thank you for the comment. We will be, and we understand the dynamic between the two. It's a very delicate balance that has to be judged and continuously judged. And so we will take your comment into consideration and we appreciate it. Just on the, you're from the Pediatric Society, and I just wanted to mention that there has been some discussion about pediatric trials, and our view is that we would like to start pediatric trials, at least in 12 to 18-year-olds, as soon as possible. And then based on the safety and immunogenicity that we see, get down into the younger age groups as well, because we think pediatrics is a very important population to consider vaccination, with safety being a very important consideration as well.
Dr. Kimberlin.
One follow-up question along obviously the same lines. How many subjects were enrolled before the halting event occurred in one subject?
I don't have the exact number of how many were enrolled at that time. We might be able to get back to you on that. I just don't have that number.
Would you speculate that it was many thousands?
No, we've just started the trial in the last month in September, so we don't have many thousands of subjects. That's correct.
Dr. Maldonado.
Thank you very much. Bonnie Maldonado, Chair, Committee on Infectious Diseases for the American Academy of Pediatrics. And I also wanted to voice very strong support for in particular pediatric transparency. I recognize that children under 18 are considered tier 3, but it's critical during this time, in particular, given the sentiments around vaccines in general and the downstream impact of vaccine confidence in all of our immunizations that these particular trials will have on our pediatric populations and our ability to control preventable infectious diseases. So we really urge this group to be as transparent as possible. Families are concerned about primum non nocere. They really want to make sure that their healthy children are going to be protected, not only from this devastating illness, but from others. And we really need to have every single family confident, and every pediatrician in particular confident in this vaccine. And we know that the populations at highest risk are not children, but if we want to reduce transmission, which is the ultimate goal, and achieve herd immunity through vaccines, we're going to need to have pediatric populations engaged. So this is a critical group. They know how to vaccinate, as you heard earlier from our colleagues at the AMA. Pediatricians do this all the time, and they really depend on the Academy and the CDC in particular for their guidance. And that guidance is always dependent on transparency. Thank you so much.
Yeah, we plan to go into children as soon as we possibly can, but very carefully in terms of safety.
So, Dr. Sadoff, thank you very much for answering our questions. Thank the voting members and the liaisons for posing them. We're going to take a quick 5-minute break here. We will extend into the lunch hour or lunch 45 minutes, so lunch will be a little shorter than had planned. Let's have everybody come back here at 13 minutes past the hour. Thank you very much.